project. Today, I'm going to be talking about the impact of sex on brain development and radiation response. I'm going to start off with a well-known paradigm of sexual differentiation. And we should all know that sex differences fundamentally stem from a difference in sex chromosomes. So if you have XY, you end up developing testes. If you have XX, then you develop ovaries. And it's these gonads that secrete hormones such as testosterone or estrogen that could wash throughout the body and cause sexual differences. And this is all true and is responsible for many sexual differences that we observe, but it doesn't exactly tell the whole story. And so a more complex picture was developed in the 21st century or so, where yes, you have the impacts of hormones causing sexual dimorphisms, but it turns out that sex chromosomes themselves can influence or can generate sexual dimorphisms without the influence of gonads whatsoever. And so this might not be intuitive for most people, but this could all be explained by looking closer at the Y chromosome, where on this Y chromosome, there's this S or Y region, which determines whether you develop male gonads, so testes, if S or Y is present, or if S or Y is absent, then you will develop female gonads, so ovaries. And so interestingly, if we move this S or Y sex-determining region onto an autosome chromosome, then that enables us to study the effects of sex chromosomes separately from the effects of sex hormones that are secreted by the gonads. And that's what's done here in this mouse model known as the four-core genotype model. And so here we have a female XX mouse and also a male XY SRY mouse. But notice the minus sign here representing how the S or Y region has been moved onto an autosome. And so if we were to breed these mice, we have four sex genotypes. We have, again, our sex XX and XY SRY, which are our, say, traditional female and male mice. But you also have an XY mouse with ovaries or even an XX mouse with testes. And so this model is useful as it enables us to study the effects of chromosomes and hormones separately. It has been employed various times to investigate a variety of different phenotypes to investigate sex differences. So here I'm showing a table from a review that lists some of the phenotypes that this model was used to separate out the chromosome effects from the hormones. And some of these phenotypes include autoimmunity, behavior, brain morphology, hypertension. And at the Mouse Imaging Center, or at least from the previous talk given by Brian, we look at late effects of cancer treatment, which includes chemotherapy. But in this project or in this talk, I'll be focusing on radiation. And so a major question that we're trying to answer is, why do females have worse cognitive outcomes after radiation therapy compared to males? As one of the main risk factors after pediatric radiation therapy is being female. So previously, one of our PhD students investigated this and found major sex differences in knockouts of CCL2, which is a major driver of neuroinflammation after radiation. So in her paper here on the right, we're showing color maps where females do worse than males, as here they're represented in blue, showing volume loss, compared to males, which showed a volume recovery. Therefore, males respond better to radiation compared to females when CCL2 is removed or deleted. And so to summarize what we know so far, we know that males have better brain outcomes after radiation in CCL2 knockout mice. And we also know that sex differences are attributed to either hormones or chromosome effects. And this leads to the two research questions that I want to ask is one, how do chromosomes and hormones influence the developing brain? And two, are sex chromosomes or sex hormones responsible for the better outcome that we observed in male CCL2 knockout mice after radiation? So to answer this question, we need a lot of groups and a lot of mice. So just kind of steal a page from my advisor. Say that we have about 15 mice per group. And we have 2 groups for radiation and sham, 4 groups for the different sex genotypes and CCL2 knockout and wild type, as well as 5 time points because we're looking for development. And since all these mice are, since this is all multiplicative, it's 15 times 2 times 4 times 2 times 5, it requires over 1,000 scans, which is much more practical and achievable if we could do multiple mice at the time using MRI. And so similar to what my advisor presented, this is the system we have here where we could scan 4 mice at once and the scan time takes about an hour. And so we can get high enough resolution at 75 micron isotropically to discern individual brain structures. So putting this all together into an experimental timeline, so we have our 4 sex genotypes that are the CCL2 wild type mice. And since we're interested in the knockouts, we have our 4 sex genotypes as CCL2 concluding these CCL2 knockout mice. Next, we scan at a baseline at postnatal day 14. At day 16, we irradiate the mice or give them the sham treatment, and then we can scan at subsequent time points all the way up into P98. And once we have the images, we segment the volumes using our automatic pipeline in which we can analyze the volumetric data using our linear mixed effects model. And so then now we could start to answer our research question. And the first one being, how do chromosomes and hormones influence the developing brain? So we're going to be looking at a well-known structure that's canonically sexual dimorphic, the medial amygdala, and see how hormones and chromosomes can influence volume. So we have these 4 sex genotypes, which are represented as these 4 colored lines. And here I'm plotting the relative volume of the medial amygdala over time. So this is just the medial amygdala volume normalized by the whole brain volume. And you see immediately that there are 2 separate groups here, and these 2 groups represent the mice that have SRY, so these guys on the top, and then these guys on the bottom are the mice that don't have SRY. So these mice have testes, these mice has ovaries. And this is this is consistent with what we know with males having larger medial amygdalas compared to females. But if we look carefully within each one of these branches, we can see that there's further division within this chromosome effect. So the mice on the bottom here are the XY mice, whereas the mice on the top are XX. And so this is interesting because male hormones tend to, or at least for the medial amygdala, result in larger volumes, whereas male chromosomes result in smaller volumes. And so this is an example where these systems can compensate for one another, and it demonstrates the observational power of having this model in order to separate out the sex chromosomes and hormones effects. Next, I'll be showing the brain during neuroanatomical map of the brain, so other regions as well. So on the left here, I'm showing the chromosome effects. On the right here, I'm showing the hormone effects. And each row represents a time point in which these differences tend to emerge. And then the colors here represent areas that are larger in males. So those that are red are areas that are larger in females. So they're colored in blue. And we can see immediately that there's not much overlap, as it appears that chromosomes and hormones influence different structures or in some sort of uncoordinated manner. And to point out clinically, perhaps one structure of interest is the pituitary gland, which we have here, which was found to be larger in mice with female chromosomes. And this is interesting because the pituitary gland is considered to be this master gland that controls the function of other glands. And it could also hint at some interplay between hormones and chromosomes. So to answer the first question of, do hormones or chromosome influence the developing brain? We would say, I think, quite differently, as there appears to be diverse influence in some sort of uncoordinated manner. To answer the second question, which is looking at radiation, it's asking whether sex hormones or sex chromosomes are responsible for the better outcome in CCL2 knockout mice. So to start off, I'll be showing the irradiation effects. So we look at the radiation effect in wild-type mice, in traditional female mice, which I have here, and male mice, which I have here. On the y-axis, I'm showing the volume of one of the structures, the corpus callosum, over time as an example. And you can see at postnatal day 16 is when we give irradiation. And there's a volume difference between the 7 gray mice versus the sham mice soon after, as early as P23. And between the male and female mice, we see that these volume differences are quite similar, therefore not much of a sex difference. On the right here, I'm plotting the brain map showing regions that were significantly affected by radiation. And you can see once again that these male and female mice have structures that are similarly affected. Next, we're going to layer on top the CCL2 knockout mice. So once again, we have the wild-type mice between the females and males, not much of a difference. But when we add on the CCL2 knockouts, we start seeing the sex difference. As CCL2 knockout mice, females have larger volume loss compared to these males, which was consistent with our previous findings. And now the question is, do males do better here because of hormones, so the presence of S or Y, or because of chromosomes, because of this Y chromosome? So to answer this question, I'm going to be showing another brain structure with a similar pattern where female knockouts do worse than male knockouts. And so say that here we add on the S or Y region. So we give these XX mice hormones or male hormones. We see that these mice actually do better. Therefore, that we can conclude that there's likely to be a hormone effect. And what if instead of adding the S or Y, we swapped out this X and put on a Y chromosome? So we have an XY mouse with ovaries. Well, these mice also do better. So there also appears to be a chromosome effect as well. And so it appears that both hormone and or chromosome effect seems to be responsible for this improvement. And this is true for other regions as well. So here I map out the chromosome effects and the hormone effects in the brain. And again, the rows represent the different time points. And we can see that across many of the structures, we see a similar trend in recovery where these chromosomes or hormone effects have lots of spatial overlap. So to answer the second question, are sex hormones or sex chromosomes responsible for the better outcome after radiation in CCL2 knockout mice? I think I would say yes, in a way that they're quite similar and that they have similar spatial overlap. So to conclude this presentation, as well as give us some takeaways, from the first question, we can answer with sex chromosomes and sex hormones influence different brain regions in some sort of uncoordinated manner. And we did this by demonstrating novel spatial temporal maps of brain development. And hopefully this can help us further understand sexual dimorphisms in the brain and for helping understand sexual differences in neurological conditions, which may be dependent on hormones or chromosomes. And secondly, maybe more clinically relevant to radiation survivors, was that we showed that both male chromosomes and male hormones provided a benefit in CCL2 knockout mice in similar brain regions. And this can tell us that sex differences in cranial radiation survivors might be attributed to both sex hormones and sex chromosomes, and that therapeutics that mediate neuroinflammation after radiation might want to consider both these effects. So that concludes the presentation. I'd like to thank my supervisors as well as our collaborators and open up the floor for any questions.